So it gives me distinct pleasure to talk to you today about polyanhydride nanoparticles and how they can serve as a platform for the design of effective and novel universal influenza virus vaccines. So I've been asked to first introduce myself and what my laboratory thinks about it. My lab is really focused on the initiation and regulation of adaptive immunity, really to pulmonary pathogens, whether that be viruses or bacteria. And we think a lot about both the immunity to as well as the pathogenesis of those types of infections, with an eye towards understanding how dendritic cells, which are really the sensor cells in the lungs that see that you have an infection, and how those cells educate the adaptive immune response, whether that be the T cells, either CD4s or CD8s, or the B cell, an antibody response, and fight off that particular infection. How do you know that you have a bacterial infection and respond with an immune response of Y versus a viral infection and respond with an immune response of X in order to tailor that immune response to the actual infection that you have? And we think a lot about how those dendritic cells sense the pathogen, go to the regional lymph nodes and educate the adaptive immune response to respond appropriately. We think about that with regard to infection, but also to vaccination and priming better vaccines against pulmonary viruses. And that'll be one of the subjects for the day, looking at vaccination against influenza. But my lab is also interested in understanding how things like chronic alcohol abuse change the pulmonary environment in such a way that the immune response has changed, leading to increased levels of disease, as well as how sepsis alters dendritic cells, which leads to long-term exposure and increased incidence of infections. And then we also look at how bacterial and viral co-infections alter the immune response and lead to increased levels of pathogenesis. So again, my lab really thinks about how these dendritic cells talk to T cells and result in a proper immune response that's required to clear an infection or to prime an immune response that can clear an infection after vaccination. So I don't think I really have to spend too much time convincing you that influenza is an important human pathogen, and it's really going to be the topic for the day, is looking at vaccines against flu. It's a major economic burden on the US, whether that's in lost work or productivity, but about 10 to 20% of the population is infected every year, results in about 250,000 hospitalizations, and somewhere between 36,000 and 49,000 deaths in the US every year. And it's changes in these coat proteins, the hemagglutinin and the neuraminidase, that occur every year that has us to go and get vaccinated every year because of these small mutations in this coat protein, and we call this antigenic drift. But when two of these viruses infect the same cell, you get repackaging of these 8 segments, and you can create a brand new virus. And these are called antigenic shift events, and these typically lead to pandemics. So our currently available vaccines are largely the flu shot, if you will. That's a trivalent or quadrivalent inactivated vaccine. So what's quantified is the amount of HA in that vaccine, and it really drives a serum IgG response with little to no de novo T cell immune response, and really no immune response in the local lungs or the nasal passages because it's given intramuscularly. The alternative vaccine is the live attenuated influenza vaccine, and this is a temperature-sensitive vaccine that's attenuated so they can only replicate in the nasal passages, but not in the lower lungs. Now, it more closely resembles natural infection, and there's a little bit of T cell response, but not a really robust T cell response. No real immunity in the lower lungs, only in nasal passages because the virus is replicating there. And then because it's an attenuated vaccine, it cannot be administered to high-risk groups. So one of the things we want to do is try a more universal or cross-protective immune response through vaccinations, and really come up with some of these or answer some of these limitations of our current vaccines. Now, it's clear for many, many years the role of antibodies, and we'd like to drive that type of B cell and antibody response with our vaccination strategy. But nature actually did the experiment for us in 2009 that shows that T cells are also very critical after infection in mediating protection. And what happened was those individuals that had T cells that crossed against a new pandemic strain in 2009 either didn't get disease or had a very mild case of disease. Or those individuals that did not have those T cells had a very severe disease. So we would advocate that a universal vaccination should take advantage of both the antibody and B cell mediated arm of the immune system, as well as the CD4 and CD8 T cell mediated arm of the immune system. And it's this latter type of immune response that's referred to as heterologous protection or heteroceptive protection. And the current vaccines don't do a good job of driving this type of T cell response, or really lung resident type of responses as well. So biology has already talked to you about some of the benefits of polyanhydride nanoparticles. But for the sake of this talk today, I want to point out a few things. One, if we're giving an intranasal-based vaccination, this is a needle-free type of vaccination. And as we've learned over the last year with COVID vaccinations, one of the nice things is that we can break the cold chain using these nanoparticles, and they're very biocompatible. One of the key concepts that's come under understanding our immune response to a natural infection has just been this idea of tissue-resident memory responses. And I always explain this to my students in this way. If I had to talk to everybody in the room, it's much easier meeting them at the doorway as they enter the room, right? As opposed to coming in from another city or another state, which is going to take me longer to get there, and it's going to take me longer to find everybody in the room and make sure I've talked to everybody. In this case, we want to park resident memory T cell responses at the site of a local infection. And in fact, in your lungs right now, you have flu-reactive T cells when they've looked. All right. And so we want to park these cells in the nasal mucosa and the lungs with vaccination strategies. And this happens actually with an infection. You get immunity in the lungs, the nasal passages, as well as systemically. With our current vaccines, you don't get this. With the nano vaccine given intranasally, we have the ability to drive lung resident, nasal resident, as well as systemic immune responses. The other advantage of giving an intranasal vaccination is we can take advantage of the system and how it's designed to drive immune responses in the lungs. And one of this is the dendritic cells that live in the lungs and sense these types of infections imprint on the T cells when they activate them, the ability to return to the lungs and fight the infection. And so we'd like to use those DCs to instruct our vaccine-induced T cells and B cells to go to the lungs and fight the infection where the actual infection is going to occur. The other main thing that nanoparticles really confer to us is the ability of an antigen depot. And I referred to the tissue resident response just a minute ago. Well, we know that having extended antigen depots boosts this tissue resident response. And in fact, infection with flu does leave behind an antigen depot well after the virus is cleared. The nano vaccine, because of its design, can do the same sort of thing. But the two current flu vaccines do not do this, particularly within the lungs. All right, so I'm going to generally summarize what the vaccine is from a general standpoint of what we call IAV nanovax. And for the sake of today's talk, I'm going to be talking about a vaccine with an HA protein and an MP protein from an H1N1 virus, combined with a CpG sequence that causes cross-presentation. So this enhances that CD8 T cell response. And the vaccine is based on a CpTEG CpH nanoparticle-based backbone. All right. So for most of what I'm going to talk about today, it'll be a mouse model. And so we can prime with the vaccine, 14 days later, boost or not boost. And then at various stages throughout this vaccination, pull immune responses and look at what we're generating both in the lungs, nasal passages, as well as systemically. And then some of these animals we're going to go on to challenge, in this case with a homologous challenge. So if the vaccine is going to work, it needs to be able to protect against a homologous challenge. And so what we've taken is naive mice. We've challenged them with a lethal dose of homologous H1N1. And what you can see is that this dose will kill 60% of the animals. And in fact, they lose a substantial amount of weight as a measure of morbidity. And this PNH is a measure of lung function. And you can see that it goes up, meaning that they're having respiratory distress after the infection. But if you look at the prime only, now you get 100% protection, or the prime plus boost, again, 100% protection, no change in weight loss, and no change in lung function. We also observe a substantial and significant reduction in viral titers in the lungs 3 days post-infection. So we hope to drive a B cell immune response, and in fact we do when we've looked at the B cell immune response by counting the number of B cells that are in the lungs, the ability of those B cells to make germinal centers, or those B cells to class switch from an IgM to an IgG phenotype. What you can see is that flu drives a nice B cell response, so does Nanovax, and a nice germinal center response and class switching. But as expected in the lungs, LAIV and IAV do not. They dual drive a response elsewhere in the body, but not in the lungs. We look at the ability of those B cells to make antibody. In fact, what you can see here in the open squares, the IAV Nanovax drives a nice serum IgG response. IIV, or the flu shot, as expected, will drive a serum antibody response, but it's much lower than what you see with the Nanovax. And in fact, Nanovax is very similar to what you see with the actual infection. What IAV won't do is drive that pulmonary immune response. And again, we see no IgG or IgA to IAV. But in fact, with Nanovax, we can drive a nice IgG response as well as an IgA response at day 32 and day 45 post initial vaccination. Now, the proof is in the pudding. Are those antibodies reacting against AJ? Can they confer protection? And typically, when one looks at serum IgG levels against flu, and we do it by a mechanism called hemagglutination inhibition, and above a 1 to 40 threshold is considered protective. IAV will drive that type of response systemically, but so does Nanovax and IAV infection. But IAV, or the flu shot, will not drive that type of response when one looks in the bowel or the bronchoalveolar lavage, again, the pulmonary immune response. However, Nanovax, as well as the IAV infection itself, drive this type of neutralizing antibody. So one of the hopes was we'd be able to drive a T cell response on top of the B cell response. And one of the ways we've looked at this is by using what's called surrogate markers. Now, we've used conventional techniques as well, such as tetramers and ICS assays, but we like to be able to look at outbred populations. And what the way this works is you see changes in markers like 49D and 11A on these T cells that tell you that they've been antigen experienced. And this is technology developed by Steve Varga as well as Vladimir Batavinix. For CDH, you have changes in CD8 alpha and 11A. This has been validated now in mice, humans, ferrets, and non-human primates. That you can measure the response even when that population is outbred. You can measure the T cell response in the blood or other organs. All right, so showing you how this works, if you look at the CD4 T cell response, they have about a 10% background level of antigen experience prior to any vaccination. Flu mist or shot, as expected, doesn't drive a pulmonary or lung-specific T cell response, but IAV infection does, and so does Nanovax, and this is 7 days after the vaccination. With CD8 T cells, again, about 5% of a background level. Flu infection drives a very robust antigen experienced CD8 T cell response in the lungs, and so does Nanovax. And if one looks, what I showed you was day 7 in those previous graphs, Nanovax is a little bit below what you see with IAV infection. Again, LAIV and IAV don't drive this type of response. But at 32 and 45 days, Nanovax is now almost equal to or above what you see actually with the infection. Now, one of the things that I talked about early on was we wanted to drive a resident type of T cell response. And so the question is, can we do so? For CD4 T cells, the markers for this are you express CD69 and your CD103 negative on top of the antigen experience markers. And this is this red box. And in fact, Nanovax, similar to IAV, drives this resident CD4 T cell response. We're also getting what we call up here these green boxes that express 103 as well. It's unknown what these are, but in humans, some people believe that these may be cytotoxic CD4 T cells, which could help confer protection. For CD8 T cells, the resident memory response is CD103 positive and CD69 double positive, the red box again. And again, in CD8 T cells will drive a resident response to Nanovax, similar to what you see with the infection without the harmful side effects of the actual infection. So since we've gotten that T cell response on top of the antibody response, can we get heterologous protection? So what we've done here is vaccinated animals with the H1 proteins, or proteins from H1 viruses, and challenged with a true heterologous strain, so a Hong Kong 68 or an H3N2 at a severe, really robust challenge, a lethal dosing challenge, where all the animals that are naive die within 7 days. A prime only leads to 40% protection, where a prime plus boost leads to 80% protection in this really robust and stringent model. And we can see reductions in PNH with the vaccination and reduction in morbidity measured by weight loss as well. Now, you don't see this same type of heterologous protection with an IAV infection, or excuse me, the IAV vaccination. You do, as expected, get protection against homologous challenges, but you don't see this type of protection against heterologous challenges with the flu shot. So what I've had time to show you today is that we can drive really nice, robust local responses, nasal and lung, as well as systemic T cell and B cell responses for the Nanovax. We can induce resident memory responses that should increase the levels of protection, and we can confer protection in both homologous as well as heterologous exposures. Things that I really didn't have time to show you today is we've shown this now in multiple animal models, including mice, ferrets, pigs, and non-human primates. We've been able to work out a dose that would work for a single administration, giving the same level of protection as the prime plus boost strategy. And we get a very long-lived protection with this type of strategy. And again, this has worked in several outbred models, not just the inbred strains that I showed you in today's talk. You confer both homologous as well as heterologous protection whether that be ferrets or mice that we put it in, and now again in non-human primates. And I've been able to show you that we've been able to boost didn't have time to show you, but we have data that suggests that we can boost immunity and protection in pre-immune models. What it showed you was in fact in naive animals, but you would want to boost immunity in pre-exposed or pre-immune animals, or like us, we're all going to be pre-immune to flu. And in fact, we can boost that immune response with vaccination. Because of the plug and play nature of this, we can swap out the antigens. And we've actually created vaccines using different backbones of HA and NA, as well as other viral proteins from flu, and show the same levels of type of protection. We've also been able to change the adjuvants and get the same sort of protections as well. And importantly, we've been able to get protection to both seasonal as well as high path avian influenza virus infections using this strategy. So what I've shown you today has been largely work that's been a collaboration between myself, Tom Welchman, and biology. A lot of this work is published in this manuscript in Frontiers in Immunology, and has been supported by 2 R01s, as well as 3 non-clinical services agreements with the NIH. But this is part of a larger project, NIH-funded studies that are looking at nanoparticle vaccination against influenza, including folks at Mayo Clinic, University of Wisconsin, St. Jude, Vanderbilt, Sweary, and Zateo, as well as additional investigators at Iowa State. It's now being supported by 4 different R01s. But again, it's part of our larger group, and we've started to submit grants on additional components of this, including other groups such as Mount Sinai, as well as formed a company in the last year called Nanomed. The 4 principals are biology, myself, Tom, and Mike, and it's patent protected technology. And I'd be glad to talk to you more about this or other things that you may be interested in. My contact information is here, and I thank you for your time and attention. Thanks.